So the last section of chapter 10 that I want to talk about is some things that can go wrong with DNA if it's left in a single-stranded form for very long. But before we get there, let's talk more about the genetic implications of DNA structure. So now that we have, progressed enough through history and we figured out that this DNA genetic material is actually the mechanism or the biological chemical that is allowing genes to be expressed from generation to generation. There's four major things that DNA structure has to do, and the first one is that it has to encode base sequence. And so that base sequence is really setting the stage for the protein that is going to be created and expressed. The second major implication is that DNA has to be able to replicate faithfully, and we're going to hit this hard in chapter 12. This faithful replication is really made possible by the pairing of the certain bases. So in the replication chapter, chapter 12, which we're going to get to next week, is that's where we actually see the double helix actually having to unzip in order for it to be replicated. The third major implication of DNA structure is that DNA has to be able to translate its instructions from its DNA form into a phenotype. So DNA is going to be expressed through a process. And the first part of that process we call transcription. And that's where DNA is going to make RNA, or is going to be made into RNA. So that's transcription. The second process is termed translation, and this is where the RNA is going to be translated into protein code. So those two processes we're going to hit very in-depthly in chapter 13 and chapter 15 respectively. And finally, the fourth genetic implication of DNA structure is that it has to have the ability to vary. So DNA has to have the ability to vary so that it can deal with environmental stressors, pressures from the environment, so evolution can take place. So the central dogma that was first postulated by Francis Crick really comes down to three major processes. First is DNA replication, where the DNA material is actually copied into more DNA material. So think about this as like mitosis, okay, and meiosis, where we're copying a lot of DNA material. The central dogma that's centered around protein expression is going to be referring to the two processes of transcription, where DNA is going to be transcribed to RNA, and then translation, where RNA is going to be translated into protein code. So this slide right here, in essence, is your synthesis project. So you guys are going to work as groups to show that you know the difference between DNA replication, transcription, and translation. All right, so we've talked a lot about DNA and why it's so stable and how it is the biochemical component that really allows protein to be expressed from a phenotypic standpoint, but realize that there are some exceptions to this rule, and one of the big ones is viruses. So viruses are a completely different bag. They have the ability to transfer information from RNA to DNA, which is completely different than anything else we deal with. Viruses can also transfer information from RNA back into RNA. So that's what can make them so dangerous, because they can replicate so quickly inside a host cell. So during the process of replication and even transcription, DNA is going to be separated from its double helix form, and these are what we call replication bubbles. So realize that DNA really, really likes to be bonded to its buddy. Okay, it helps to stabilize it so that it doesn't start going through the apoptosis process and things don't start breaking down. But in order for DNA to replicate or for protein to be produced, we actually have to pull apart the DNA strands from one another in just a small section called a replication bubble. And so that exposes a single strand of DNA. And if something goes wrong and that single stranded DNA doesn't get stabilized in a timely manner, it can actually form a couple of these secondary special structures. So the first one is a hairpin loop, and this is where if um, is the DNA sequence is conducive, it will actually try to pair to itself to try to stabilize itself. So it forms this stem and loop structure. And here we can see on the stem that it's wanting to really bond with each other. So A is bonding to T, G to C, C to G, G to C, and T with A. So that's going to be one structure that we can see. We can also see just a stem structure where this phosphodiester bond that's holding thiamine and adenine together gets really stretched as it is trying to bond to itself. That gets super fun. And then another form that we can see is this cruciform that actually occurs with both strands of DNA. So essentially, each strand of DNA is going to form a hairpin loop. So I just want to end the chapter talking about things that can enhance DNA's ability to be replicated or actually tie it up. And this is really getting to post-translational modifications or methyl groups. And a methyl group is shown here on this structure. So this is 5-methylcytosine. So this methyl group is actually attached to a cytosine molecule. And what ends up happening is that this methylation process has evolved over time as a protection mechanism. So in bacteria, their genome actually really likes methylated DNA because it protects it from viral attack. It protects it from viruses sneaking in and taking over. Methyl groups are going to most often be bonded to a cytosine. We very rarely see them bonded to adenine, thymine, or guanine. And the degree of methylation is really going to be different depending on what species we're talking about. So we really don't see any methylation occurring in yeast. We see a small percent of methylation occurring in animals, but we see a really large percent occurring in plants. And this becomes really important because that methylation process is going to allow us to and allow plants to evolve and be able to function in different environments. This also becomes very handy when we're trying to make transgenic plants by inserting GMOs because we can methylate certain structures and get genes to turn on or to turn off. So DNA methylation is really, really important to agriculture in the crop field. And one just final note about DNA methylation, it actually has the ability to prevent transcription. So we can use methylated gene therapies to prevent like genetic abnormalities and diseases by stopping transcription from taking place. All right, so this is our last slide in chapter 10, and this really is a 3D structure of a double helix that is being, some might say attacked, but it's not attacked. It's actually being escorted by proteins. So the proteins are going to be the ribbon-like structures in red and in orange, and these can be DNA polymerases or RNA polymerases. And what they do is they actually will surround the DNA. They will help unzip the double helix so that replication and transcription can take place. And then DNA is going to be the ball-like structure in the center. We can see the green balls here are the sugar phosphate backbone. And then the blue balls are going to be the hydrogen bonds that are holding the nucleotides together. All right, so this is a good 3D image and a good image to end on. So good luck with chapter 10.